Hi, my name is Dennis Akkad. I'm VP Research at Absci and Program Lead for ABS-201. Within my role as VP Research at Absci, I'm ensuring that the antibodies which the talented AI and wet lab scientists create have a seamless transition into the clinical development. Prior to joining Absci, which was an amazing opportunity in my career, I worked for a decade in big pharma across multiple indications, spanning from heart, lung, kidney, and endometriosis in women's health. My background is a trained biochemist with a PhD in neuroscience and also a training as laboratory geneticist. As you have heard from Andreas, the prolactin receptor, ABS-201, is one of these rare occasions as a geneticist where you really have these cases where human mutation guide you with regard to the safety profile, but also with the efficacy when we're looking into the mode of action. And I'm very excited to announce that we have dosed our first participant in our headline trial. The phase 1/2a study aims not only to look into safety and tolerability as the main readout, but also we anticipate to have our interim efficacy readout in the second half of 2026. At Absci, we always go big. This is why we have designed the ABS-201 headline trial in a way where we not only combine safety and tolerability, but also efficacy in a really fast way. So how do we do that? So basically what we have thought about is how can we combine the preclinical readouts which we have from the monkey study and the ex vivo study in a really nice clinical trial design. And I'll walk you through that, how we have outlined it. The headline trial is a double-blinded placebo-controlled trial which aims to enroll up to 227 participants with and without androgenetic alopecia across single ascending dose and multiple ascending dose cohorts. Within the SAD portion of our clinical trial, where we are looking into up to 4 single ascending doses in healthy volunteers, we will mostly assess safety and tolerability, but also immunogenicity and, what is most important, pharmacokinetic profile. So how long is the half-life of the antibody? We will then transition into the multiple ascending dose portion of the trial, which has been designed in a way to be statistically powered to show hair growth efficacy throughout this treatment duration. What do I mean with that when we're speaking about treatment duration? What you have heard earlier from Andreas is that within this naturally occurring androgenetic alopecia stump-tailed macaque model, those monkeys were treated for up to 6 months, and we're aiming to do so for the healthy volunteers. So this means that the healthy participants are dosed over 26 weeks, and we will do a hair regrowth assessment at baseline, at 13 week, and then at 26 week, and then look at the hair regrowth progression over time. Based on the engineered long half-life of our antibody, we will follow up the participants for nearly a year, and within this follow-up period, we will further interrogate the durability effect of the hair regrowth. The hair regrowth assessment is measured by taking global images from the healthy volunteers, but also what Professor Paul showed you, macroscopic images of the scalp area where target area hair count, target area hair width, and target area hair darkening is measured in order to get from this quantifiable variables regulatory approved measures for efficacy assessment. Aside of these quantifiable measurements, we will also have patient-reported outcome measures, which are very important because it's one thing if you have really a numeric improvement, but it's really most important that the participant himself feels or sees that there's improvement on the hair regrowth in order to have afterwards a quality of life improvement. Of course, the next question is, when are we going to have our interims readout? As mentioned earlier, we're treating our participants with androgenetic alopecia over a period of 6 months, mimicking not only what we have seen in the stump-tailed macaque study, but also reflecting standard practice in hair regrowth trials. Within this 6-month period, we will have a baseline assessment, and then an assessment at 13 week and then 26 week. Now, when are we expecting the first interims readout across all 3 multiple ascending dose trials? That will be as early as Q3, Q4 in 2026, and the 26 readout as early as beginning of 2027. These readouts are important and will guide us in our registrational trials moving forward down the line. The described phase 1/2a headline trial will take place in Australia within the multi-site setup. And we're super excited that one of the PIs will be Professor Rodney Sinclair, who is a world-renowned expert in androgenetic alopecia, and will talk to you more about ABS-201 and the headline trial.